I'm very pleased to say we have two excellent speakers. The first one of those is Precious Mongwe, who's from CSIR South Africa, and he's going to be talking to us about climatic controls on the metabolic constraints relating to the twilight zone, of course. So with many thanks, Precious, over to you.
Thank you. Thanks very much, Adrian. Let me share my screen so that it works out. Okay. Okay, I got it. There we go. Sorry about that. Good morning, good afternoon, everyone. It's afternoon where I am. So thanks once again for the invitation to come give this talk. So my presentation is climatic controls on metabolic. I don't know if you can you, okay, I'm trying to move things out of the way. Yes, climatic control on metabolic constraints in the ocean. So this is the work that I did mostly at Anchorage, my postdoc, my first postdoc work, and I was working with this colleague. So Matt was the PI, Matt, Taka, as well as Katas on this project. So I understand I have about 20 minutes, so I'll just get going. Maybe when I hit the 20 minutes mark, just if I'm not done, please just give me a notice, just a hand so I know. Thank you. So, okay, so I think to give a constraint, so essentially by constraints here, essentially we are focusing, using the, looking at the metabolic index in the context of oxygen and temperature constraint. So let's see if I can move slides, not yet. Okay, there we go. Okay, so maybe I start here as a way of sort of framing the introduction by saying oxygen is essential for organisms. And I appreciate how obvious the statement is, and we almost don't need to hear it. But I think it's important in the context of that the ocean is not homogeneous. Oxygen concentration in the ocean is not homogeneous. And this is illustrated here by this figure. So in the top figure, we have the volume, ocean volume, and then in the bottom, you have fraction of species. And then in the x-axis, you have the oxygen concentration. So we see that oxygen have a different distribution in different parts of the ocean, and that as well sort of gels with how many species can survive under different ocean conditions. So the idea here is that although oxygen is abundant in the atmosphere, because of the differences in the concentration in the ocean, that even if it can change slightly, it will essentially have an impact on the organism's habitability. So it will have an impact on that. So the idea is that if it increases slightly or by margin, or increase or decrease, it also may allow more habitat or decreases habitat. So the focus of the study was try to understand how is oxygen changes in the context of warming impact organisms in the ocean. And as you realize that we are not really worried so much about the surface ocean because of oxygen really equilibrates very fast. In the context of oxygen, we worry too much at depth. In the surface ocean, the impact is much more warming than oxygen, but that will become clear as we unfold. So maybe to set up again the sort of high level mechanisms, mechanistic understanding of the drivers of oxygen. So here I have a schematic that just give a very high level broad brush of the mechanisms that circulate oxygen in the ocean. So essentially on the surface, the ocean mixes very, very quickly. The surface ocean equilibrates quicker with the atmosphere, as I said, but how is essentially oxygen transported to depth? It essentially mixes usually at the high latitude where you have very cool surface ocean and very deep mixed layer depth that allow the ventilation that transport oxygen into the deep ocean. And in low latitude is very warm because of solubility of oxygen given up into the atmosphere. And in the surface ocean, you have a consumption of oxygen, you have a production of oxygen, excuse me, by primary production, and as well as you have the consumption at depth. So by this, essentially, we have what we need for this talk. There are details, but I think that with that, we have enough to proceed. So moving further, so why are we concerned about this? So I think the angle that we are thinking about this, or that we were thinking about this, is in the context of deoxygenation. So to illustrate that, I have this figure here. So in the x-axis, you have temperature on the left, and the y-axis, you have oxygen change. And so this is the contemporary period, or the historical period. And you have the different lines here are different CMAP5 models. The black line with the gray shading is this CSM large ensemble, CSM1, which is what I'm using for the study. So we see that as the ocean get warmer, there is an oxygen loss that we do see. But, we do expect this anyway, that as the ocean warm, we do know that by solubility, the ocean degasses a lot of the gases. But if we calculate the solubility component alone, as shown in the black line here, we see that actually models shows an oxygen loss that is that's greater than the temperature effect alone. So we do know that the loss that we see, it's not only due to warming, but there's also a circulation as well as a biological component that's involved. And then we have another problem here, which, we don't know the answer to, but is that when you look at this silver or yellow line here, these are the observations estimates. So when we look at the oxygen loss with warming, it's much greater in the observations than it is in the models. And so this was CMAP5 models, but as far as I know, in CMAP6 we still have the same problem. And we don't know why this happened. So I think the idea is that we are underestimating oxygen in the models. We've been thinking about this, but I don't think we have a good solution as to that. Is an active research, which I'm not talking about here. And then we do know that going to the future, as the ocean continue to warm, that we will see an oxygen change. So the context is that because you are losing oxygen and you are warming, you are likely to impact organism distribution at the very least, if not losses of habitat. And the context in which we think about it is from the metabolic context. Here, so here I have an illustration to illustrate that. So on the y-axis, you have the metabolic rate, or the locomotion rate. In the x-axis, you have water temperature. And here we have the two kinds of organisms. You have endotherms, which can essentially regulate their own temperature. And then you have ectotherms, which is ectotherms organisms, which is, I'm primarily talking about ectotherms organisms. These are organisms that their metabolism is influenced by the external temperature. So if the temperature is very high, you have a higher metabolic demand, and when you have lower temperatures, you have a low metabolic demand. So the idea is that as temperature increases, you'll have a higher metabolic demand. So that you require ATP to make energy, so you have more oxygen. So we can see already that there's potentially a problem. There's deoxygenation with oxygen loss. So. And the framing in which we think about this is the last picture of the framing component, is that we think about this in the context of natural variability, because we do know that as much as the scenarios that we have of the future are very smooth, the ocean is rarely a smooth environment. We see that when we take the global average. But here, for example, just to make an illustration here, we have surface temperatures in air temperatures in different parts of the US. So if we have Moseltown, Seattle, as well as the whole United States and the globe. So we see that, for example, if you compare Moseltown and Seattle, you see that in the black line is the natural record, is the record from the observations. The blue lines are the coolest ensemble. The red is the warmest ensemble. So we do see that, for example, at Moseltown, that organisms that live in Moseltown maybe can survive about a half a degree variance in temperature, right? So if temperature vary within those constraints, within the range, they are fine. But in Seattle, you have a larger range, right? You can even go up to 3 degrees almost. So you see that. So this is how nature is happening. But as the ocean gets warmer and warmer, the idea is that the surface ocean changes may go out of this range. And that's why then you start to begin to have an effect into habitat. So we try to think about this in the context of when does this changes emerges, and we use the time emergence kind of approach to try to solve this problem. So this is the math slide of the metabolic theory. So this is, I'm going to use the classical metabolic theory, which it's been around for a while, actually. In the late 40s already, there was some papers looking at this, but it has been sort of revived in recent years, particularly at the Santa Fe Institute. But Katas Deutsch also has put up some papers in recent years. So the idea here is to look at it, the context of it is to look at oxygen supply and demand. And you have the oxygen supply represented by, represented by this equation with the, the body mass. You have a body mass and the partial pressure of oxygen supply, and then represented by this equation, which is sort of an Arrhenius function with the energetic energetics there, as well as the diffusion. So it's trying to think about the supply of oxygen, how temperature affect the diffusion of gases, and that particular, in this particular case, being oxygen. So, and having as well as the temperature and the activation energy. So both the supply as well as the demand is coded by this Arrhenius function. So if you divide the supply by demand, then you can sort of simplify life a little bit, and then you have what we use the metabolic index, which was what came from Katas Deutsch's work. And then here you have the equation that sort of put this all together in the metabolic, use the metabolic index equation. So this equation was originally calculated for habitat on the basis of survival, but we made a slight adjustment in this particular paper to look at, we have this what we call phycrate, which is the minimum oxygen that is required for organisms to be active, so to be able to reproduce and live. So we calibrate the physiological oxygen demand to the minimum oxygen that the organism needs to survive. So here, habitable means that an organisms can reproduce as well as move, not only they survive. And this is all graphically represented in this figure here. On the left, you have in the x-axis, you have temperature, and the y-axis, you have oxygen, PO2. So below this curve, so you have a species tolerance curve here at phycrate, and essentially below this curve, organism cannot survive, and above this curve, we can survive. So I just say phi prime in order to distinguish between habitable and reproduction. All right, so I think we have all that we need. So then we're gonna get just to the physiological constraints. So essentially we have two physiological constraints that we used in the metabolic index. One is the x-axis is temperature sensitivity, which is essentially the sensitivity of the organisms to the sensitivity of metabolism to temperature. As you go to the right, you have organisms that are much more sensitive. So essentially much more sensitive to temperature. In other words, that as temperature rises, they react, metabolic rate or metabolic demand increases rapidly. And then you have also some negative values here, where you have some organisms that essentially have the opposite behavior. And in the y-axis, you have what we call the ecological hyperoxic tolerance, which essentially means is the ability of the organisms to metabolize at low oxygen concentration. So as we go high, you have organisms that can metabolize at very low oxygen concentration, but you have oxygen here at the bottom. They are very, very temperamental. They need a lot of oxygen to metabolize. So to try to graphically illustrate this, I subsample this figure into 4. You have into 4 percentile, you have low oxygen, low AC or low hypoxic tolerance, as well as low oxygen, low metabolic temperature sensitivity, as well as you have high temperature sensitivity, low temperature sensitivity and high AC. And here you have high of both, high of AC and high E0 and low AC. So you're gonna see this subsampling throughout the image, throughout the presentation. And then we have I just put 4 species, about 3 species, just to illustrate what these species look like in the ocean. So you have in the middle, you have something like a copepod that represent the middle. You have like a sort of a fish that's somewhere there. And then we have scallops and so on. And this is the curve that actually represent the metabolic demand, as well as the species tolerance curve for this different representation. So maybe just to pick one here, the 2 examples. So if we take low oxygen demand, low AC, low hypoxic tolerance, as well as low energy, low temperature sensitivity, you have the blue line. So you see, it's very high. Essentially, the oxygen organisms can only survive above this curve. Below this curve, they cannot survive. The reason why it's bell curve like this is because when you get to lower temperatures, you start to have oxygen transport or oxygen transfer being limited by molecular diffusion. So in other words, that in order to transfer oxygen, organisms need energy, need to use energy to get the oxygen in because of it's too cold. And this you end up with an effect, for example, in the Southern Ocean, where when it gets warmer, you have organisms that survive better because they can actually transport oxygen at low cost. And then when we take the orange line here, the green line where you have high temperature sensitivity as well as high hypoxic tolerance, you can start metabolizing at very low oxygen concentration, and you have a sharp rise in the metabolic demand with temperature. So that's just the illustration of what that looks like. Now we can look at how that looks like in the actual ocean. So here we have, in the bottom here, you have the World Ocean Atlas oxygen as well as temperature diagrams. And then in the Pacific Ocean, and you have the same figure that I've shown before. And now here is sort of the illustration of the metabolic index. Essentially, where it's clear, it means you cannot metabolize. The organisms cannot survive there. And when you have color, that's where it survives. So starting here from the bottom, because you have very low AC, you require quite a lot of oxygen. So there is not organisms here cannot survive in most parts of the
Okay, we've clearly had a bit of a technical glitch connecting to Precious, so if you can bear with me a moment, and hopefully Precious can rejoin and we can restart the talk. Precious, I can see you're back.
I'm sorry, I don't know what happened there. It just broke.
We lost you, I would guess, probably about 30 seconds or so ago.
Yeah, sorry about that. Okay, I'll just share and just continue from where I was.
Yeah,
sorry about that.
you.
I don't know
no
what
problem,
happened.
no problem. These things happen.
Yeah. All right, let's see if I can find the screen. I think that's it. Can you see my screen or no?
Yes, I will do in a moment, I think. It's about to. It's saying that you've started screen sharing and double click to enter full screen mode.
All right, sorry.
There we go.
Okay,
Yeah,
can
I
you
can
see
it
the
again.
screen?
Can see it fine. Thank you.
Okay, sounds good. Okay. Let's do that. Okay, so I think I was finished with this part. I was illustrating the organisms that with the representation of what the organisms look like and what these fields look like in the ocean. So with that, just a quick brush through of the tools. So I'm using the large ensemble, the first TSM large ensemble, which is so compared to the World Ocean Atlas data, the model is reasonable. So you have the temperature, oxygen, and as well as the metabolic index estimation, and this is the model. Essentially, it's just the model. The only one thing is that the model slightly overestimate the OMZs, or the low oxygen areas, that are quite intense in the model. But other than that, it's a reasonable representation of this of temperature and oxygen, which is what we need. All right, so this how then we do the analysis. So if you imagine that I have about 69 species, physiological data for about 69 species. You have temperature as well as oxygen concentration. The partial pressure of oxygen. You put it through the metabolic index framework with the temperature and oxygen fields, and then you have the distribution here of the species. So this is the number of viable species. So what we see immediately, so I drew the three different discs to represent the surface, the pelagic as well as the mesopelagic region. So we see that the organisms are most abundant in the subpolar regions, which is where essentially it's a sweet spot because it's not too hot and you have an abundance of oxygen. And the tropics and where you have OMZs, this is where you have a lesser and lesser organism because of you have a very low oxygen concentration. So only very few species can survive there according to this data set. Okay, so now let's look at the long-term change. So first thing is that we see that what happens when we compare the future and the present. So in the future, the top line is the future temperature change. I think that most of us were familiar with that the ocean warms a lot more on the surface and less at depth. But when we look at oxygen in the bottom depth, which is not, that bottom figure is less common, where we see that in the surface ocean, there's not very much changes in oxygen, except for the Arctic, where you lose all the ice and it's cooler, so you have a little bit more oxygen. But essentially, you have more oxygen loss at depth than you do at the surface. So this is the idea. And then, but we are particularly interested in the change in oxygen with respect to temperature. So to try to represent that of how essentially the forced trend or how the forced trends affect the oxygen temperature relationship is to, here I do a regression. Excuse me, something keep moving my slides. I don't know whether it's there. Okay. So here you have the regression of temperature and oxygen in the natural climate at the top. And then the bottom, you have the future, which is like really the first trend, which is the beginning of the 2020 to the end of the century. So in the surface ocean, there isn't much change except for the Arctic Ocean. But when you go to about 200 meters, you start seeing that actually in the tropics, as well as in the Pacific Ocean, there is a change where in the present climate, or the natural climate, you have a positive relationship where the changes in oxygen occur with temperature. So that's good for organisms because if you lose oxygen, even if you lose oxygen, there's a temperature decline. That means there's a compensation so species can survive. But in the future, it's the other way around, where you either lose oxygen while temperature increases. So we begin to see that the blue areas is where we expect the largest impact of habitat. So, and now here, so now we can actually look at the actual habitat on the top line, and the top figure, you have the actual metabolic index for the median species. And then we see that at 50 meters, we see the largest loss of habitat is in the tropics, in the tropics and subtropical region, which is where the ocean warms the most. And this is mostly a temperature component. So we see that the metabolic index increases in polar regions. And this is, as I said, that you'll see that it's actually because there's a little bit of an increase in the energy that is required to metabolize because the ocean is less cooler. So it's the species that cannot survive very well in the present, but it's easier in the future. And then in the bottom line here, you have the total number of habitat. So we see a lot of loss in the tropics close to the OMZs. But when you go to depth, you start seeing a lot of loss, for example, in the Pacific, where we see there is a change between the relationship of oxygen and temperature. Okay, so now we can start looking at, to try to summarize this information. So here I show temperature in the x-axis. In the left, you have temperature in the x-axis. And the y-axis, you have PO2, and then projected into the figure is the number of habitable species, just to illustrate what these changes look like. So the blue line, the blue spheres is the joint distribution of oxygen and temperature. The red is the future joint distribution of oxygen and temperature. So we see that here, this is the Arctic, the Southern Ocean, the tropics, here the Central Tropical Pacific, as well as the Northern Pacific. So we see, for example, that in the Arctic Ocean, the blue dot become red. So the organisms are actually, the habitability is moving toward more habitat. And the same thing in the Southern Ocean, that things are moving toward the right to slightly warmer and a slightly more comfortable space. Whereas in the tropical region, you have a loss of organisms. So you are moving to the right and you are losing more habitat. And this is the sort of the final graphical representation, which is, this is only the surface and all the depths are essentially here. So this is much more clearer to illustrate easier.
You've had 25 minutes.
Oh, 25 minutes. Okay. So I think maybe I can just wrap up. So finally, maybe the last two slides. So this is the variance in the ocean of the natural variance of temperature, oxygen, and the metabolic index. Maybe I will leave, because of time, we'll leave the details from it. I can just show the next slide here of how we look at the time of emergence. So here we have temperature, oxygen, metabolic index of the number of habitable ecotypes, number of species. So essentially I pick a dot in the Pacific where we see that as the ocean become warmer, as the ocean become warmer, there's an oxygen decline. So essentially we see that the metabolic index emerges both is earlier than both temperature and oxygen, because of as you increase warming with loss of oxygen, you're advancing the time in which organisms start losing habitat. And this is the final slide of the total of the showing when the emergence of this habitat changes will occur. We see that for example, the oxygen emerges earlier in the surface ocean, which is, we do know that most of the habitat change are due to temperature in the surface, and oxygen emerges much earlier at depth because of that's where we see the largest changes. And we can see that most of the changes at depth is actually because of oxygen change. So I stopped there. Sorry for going over time. This is the summary of the presentation. I won't go through it, I think, because I talked to most of these ideas. I'll be happy to take a few questions in the time that we have. Thank you.
Excellent. Thank you very much, Precious. Over to the audience. If anybody has any questions or comments for Precious, as I say, there's a lot of fascinating stuff there. So if people have any comments, then either just please say or type something into the chat. Got time for at least one question, I would hope. So Precious, can I ask a question?
Sure.
The last slide you showed in terms of the time of emergence. So it's interesting that the number of ecotypes, so that the signal seems to lag a little the metabolic index. Is that because there is a different signal to noise, or is it because it takes a little longer for the consequences of the metabolic index to change for it to have an impact on ecotype numbers?
Yeah, that's a very good question. So I think there's two things. One is that the metabolic index is representation of one species that is at the center. But when you start accounting for all species, you have a huge range. So that there's that you have when you're considering all species, the median species is not actually quite representative of how the entire system changes. So that's just the one effect. But also because of the system, as you can see, that is a bit stripy, because when you count the number of habitat, we have to count in whole numbers. So that has also a slight effect into that distribution. Yeah.
Excellent. I was curious about the stripes, so thank you for saying that as well. Yeah. I'm aware that we're sort of coming up on half hour. If anybody has any sort of quick comments or question, we could just about slip on in, because I'm aware that Precious had that technical hitch. So if anybody has time for one more question, has another question, I'm sure we can slip just one more in. I don't see anything. So I'm sure Precious would appreciate it. If you have any questions you'd like to raise with him, please just sort of contact him by email, let him know. And say we'll be putting the talk up on the YouTube channel, and I'll put a link to the paper as well. So if anybody wants to look into it further or ask Precious any questions, then you are very much encouraged. So thank you once again, Precious.
Thank you. Thanks so much. I'll stop sharing.